Antony Burgess, Ashley Dunn and Nicholas Gough were the scientists who first purified and cloned GM-CSF, patented it, and continued developing the biology of GM-CSF to guide current and future clinical uses of this agent. The process started in the early 70s, when Professor Burgess spent a decade working through the technically challenging task of purifying to homogeneity the minute amounts of GM-CSF present in mouse lungs.
My colleagues and I really believed in the fact that it was a single protein that stimulated the production of the white blood cells. The trouble was that it broke up into several different classes and it spread itself out, so we could never really get enough of it to work with. Fortunately, a new technique was invented in California. Actually got the sequence of our protein, and we were able to start the molecular genetics from there on.
In 1982, doctors Dunn and Gough teamed up to build on Burgess's work by cloning the GM-CSF gene.
The biological source of GM-CSF at that time was lung tissue, which in fact is quite a poor source of GM-CSF. So there wasn't going to be very many clones representing GM-CSF in our libraries.
It was essentially looking for a needle in a haystack. We had molecules that were very, very rare, and our job was to try and clone the genes that encoded these molecules.
Gough, Dunn and Burgess went about their research with enthusiasm and a flair for problem solving, which buoyed their team and allowed for sustained progress. The biological source for making the cDNA library was critical due to the likely very low abundance of the cognate messenger RNA. Fortunately, the library they constructed was superb, and indeed, 2 of the 100,000 clones screened proved to encode GM-CSF.
It's had a profound impact on cancer treatment. GM-CSF in fact represents the first drug based on Australian intellectual property, and it was one of the very first drugs of a whole new breed of biotherapeutic drugs based on deploying the body's own genetic blueprints from which to make a drug.
Their research unequivocally established that GM-CSF was a specific regulator of white blood cell production, and the success of their research changed the field of hemopoiesis forever. Several companies and clinicians recognize the potential of GM-CSF as a therapeutic agent to assist patients with low white blood cell counts, especially after bone marrow transplantation.
We'd actually purified a protein from mice, but we wanted to claim, of course, the use of the protein from humans. So the company actually patented the mammalian gene. That was sufficient to then allow biotech companies to say, well, we've got enough protection to take it forward as a medical product.
Building on the early findings, Dr. Dunn later used molecular genetics to explore the role of GM-CSF in physiological systems and engineered both mice making excess GM-CSF and mice making none. Analysis of these animals connected GM-CSF to tissue inflammation and revealed its essential role in the normal clearance of surfactant from the lungs.
If you don't have this molecule, then the surfactant isn't cleared and it builds up and your lungs fill up with this gluey sort of stuff, and the body has no means of getting rid of it. Absolutely, totally unexpected, but a very exciting result, and one that now has led to the opportunity of using this molecule as a means of stimulating white blood cells in lungs in order to clear surfactant.
The results of the leading-edge research by Burgess, Dunn and Gough have changed our knowledge of blood cell production to such an extent that new rational interventions in major diseases from leukemia to arthritis to cancer can benefit from the use of the CSFs. They are to be commended not only for making the initial discovery, but also for defending their patent to ensure that GM-CSF was developed clinically and that the royalties could be used to assist medical research.